I'm Carrie Miceli. I'm back to tell you about the exciting race between two companies for the first approved exon skipping drug to treat Duchenne. Now, the Duchenne patient population is amenable to exon skipping differentially depending on what the mutation they have. The most popular mutations can be rescued by skipping exon 51. In fact, 13% of the boys could all be treated with the same drug to skip exon 51 and put the reading frame back in place, enable the expression of a functional protein. What you can see, though, is once we accomplish this with exon 51, we could rejigger the Morpholinos or the 2-O-methyl oligonucleotides to target other sequences, such as exon 45, exon 43, exon 44. And you can see that as you do that, you would pick up additional populations of boys. And once you made it through all of the single exon skipping possibilities, you in fact may impact up to 80% of the boys with this sort of designer strategy of skipping the exon needed for a given mutation to render the message back in frame. So exon 51 is the one that two companies both started with because it's the most abundant and it would be the easiest to get the patients into the trial, and you could serve the most people with this first drug. And what you can see here is how such a drug might work. As you remember, exons are juxtaposed next to one another during the process of RNA splicing, and in the case of a deletion of exon 50, exon 49 is juxtaposed next to exon 51. Those break the reading frame and lead to the loss of a dystrophin protein. However, when you administer an antisense oligonucleotide that blocks the splicing process, you can force the exclusion of exon 51 such that now exon 49 is juxtaposed next to exon 52. They fit together, you've rescued the reading frame, and hopefully you could make a functional protein if abundantly enough expressed would lead to a Becker-like phenotype. So I mentioned there are two oligonucleotides in trial. One is the Morpholino, and it's the basis of the drug eteplirsen from Sarepta, aimed at skipping exon 51. It's neutrally charged, which is considered a benefit because that helps minimize off-target effects and, in fact, may contribute to it being less immunogenic. It has an excellent safety profile, and that has enabled them to dose this drug at levels as high as they can make. The other drug is known as drisapersen. It is based on 2-O-methyl chemistry. The 2-O-methyl groups are negatively charged, and that helps with solubility, but comes at the cost of potential immune stimulation. This is being developed currently by Marin. It was originally developed by Presensa, and you'll see that as it went through trial, it was picked up by GSK, dropped, and then bought by BioMarin. So the name is sometimes drisapersen, but the name from Presensa was different. Short-term phase 1 trials were what started this all off, and you could show that there was induction of skipped RNA using PCR, dystrophin, immunohistochemistry, and Western blot. This was 12 weekly injections, and there was no extension, but it served a really wonderful role of establishing proof of mechanism because, in fact, both protein and message was rescued in the muscle to some degree. And this prompted them to move forward with a phase 2 study that ultimately rolled over to extension and will be the basis of the data that's being put forward to the FDA to ask for an accelerated approval for Duchenne. So the study design was that there were 3 cohorts, 4 in each cohort. 2 of the cohorts got a different dose of the drug, either 30 milligrams or 50 milligrams, and the third cohort got a placebo, at least up until week 48, week 24. And at week 24, all the patients were rolled over to drug and have been then maintained on drug now for 4 years. The first paper published was on the first 48 weeks, and then as the boys continued, then there were publications where they reported as late as 168 weeks, and in the context of that study, they were able to find a very well-matched natural history control, which had become a bit of a sore point. These boys seem to be doing very well relative to what we expected, but how can you prove that if they didn't continue in a placebo-controlled manner? And the solution is to find an excellent group of natural history controls and compare to that. So in the first few studies, they looked for dystrophin. They did a muscle biopsy at week before treatment, at week 12, and again at week 24. They stain the muscles for dystrophin and they look for mRNA. They saw some nice RNA indicative of skipping, and they also saw dystrophin staining on immunofluorescence. However, there's some issue as to whether or not that was done in a controlled enough manner, and the FDA actually asked Sarepta to have these biopsies rescored by independent pathologists. And just very recently, they provided the FDA with the requested experiment and showed that, in fact, that all of the patients on 30 or 50 mg per kg ultimately made dystrophin protein, and there was no significant change from baseline in placebo. The interesting thing is at 12 weeks they were unable to see any dystrophin expression, whereas by 24 weeks there was a sufficient enough dystrophin to be able to observe it on a slide. And so this led people to believe that if there's going to be an effect of the drug, it would have to be after the protein made at about 24 weeks. And here you can see the protein at 24 weeks is in fact expressed. It rims the sarcolemma. It's where it's supposed to be. They've shown, in fact, that it rescues other members of the dystrophin glycoprotein complex. So this is exciting. We put in an antisense oligonucleotide systemically. It's circulated through the whole body, gotten into the muscle, into the nucleus, and forced the exclusion of an extra exon such that when the protein is made, you have an in-frame protein that has enough of functionality that it can make it to the sarcolemma and rescue the rest of the complex. And here is the result showing that that is in fact statistically significant. When it was in fact scored by all 4 pathologists, they came up with a similar result, and that is that at week 12, not much dystrophin was being made, but by week 24, you could see a significant amount of dystrophin being produced in these boys' muscles. Unfortunately, 2 of the boys went into wheelchairs prior to the 24-week mark. So in order to continue the study, they needed to exclude those 2 boys for 2 reasons. One is it's thought that those boys could not have made dystrophin before they went off their feet because they were rapidly declining before the protein was even made. And so and the second reason is that the outcome is a 6-minute walk test. So once boys lose ambulation, they can no longer be included in the main population. So Sarepta was forced to make a modified intention to treat population and to examine just the 10 patients who were walking to see if, in fact, the drug showed some efficacy. And what you can see in the early studies is that the boys that received drug stayed stable for up to 96 weeks in this slide, whereas the boys that were in the original placebo group declined 70 meters in the first 36 weeks. But the very interesting thing is, since they were rolled over, they got drug at week 24. And what you can see is the trajectory of their progression has now changed. They were on a trajectory to lose ambulation, and with treatment that leveled out and they stayed stable throughout the entire time. Now, I mentioned that as time went on, the FDA was very interested in having a nice control. There was no placebo after 24 weeks, and so they searched through the literature to try and identify a natural history control that was suitable. And to this end, they looked through 2 studies of over 200 patients and were able to cull the samples down to patients that had the same walk times, the same mutation, and in fact came up with 13 boys that are very well matched. And when you compare them in the yellow line with the eteplirsen treated boys, you can see that while the natural history controls declined by 45% over time, and then ultimately 74%, the Sarepta boys were relatively stable, losing a little bit, but not nearly as much as had been expected. And that continued through week 192. The other thing is that now that the boys have been on the drug for so long, you can ask about what's the reasonable likelihood that this protects boys from losing ambulation. Now we have to include those first 2 boys if you're going to use a measure of loss of ambulation, and so the Sarepta boys cohort lost 2 boys over that time period. But in that same time period, the natural history lost boys, 6 of them lost ambulation. So you're moving from a 17% loss of ambulation to a 40-something% loss of ambulation. So that shows that perhaps the drug is having some efficacy, preventing boys from losing walk distance and delaying their time to wheelchair. I mentioned that the FDA was very concerned about the dystrophin quantitation, and so they had given Sarepta criticisms as to what they didn't like about the dystrophin quantitation. And so the boys agreed to a 4th biopsy. It was a voluntary biopsy, but in this way they could process the biopsies exactly as the FDA had recommended. In fact, the FDA held a workshop where all the dystrophin scientists got together and said, this is the way it should be done. And that led to this protocol that was then used and scored by 4 independent pathologists again. And what you can see is that the treated boys, in every case, made dystrophin, whereas the untreated boys did not. Similarly, if you did Western blot to ask, is a functional protein of the proper size being made, they could see that in 9 of the 11 boys that volunteered for the 4th biopsy, there's a dystrophin band indicative of rescue of dystrophin protein at the proper size. So with that data, Sarepta wanted to go forward to ask for FDA approval, but the numbers are incredibly small. 12 boys is not a big study, but Duchenne is a very rare disease. And fortunately, in 2012, Congress enacted Padishah, which is the Food and Drug Administration Safety Act. And that changed a lot of things. But in our case, the most important thing is that it allowed for accelerated approval for rare disease. You know, back in the time of HIV, the folks that acted up, what they got from that is accelerated approval. And that's what drove that field forward. After that, the cancer folks petitioned and got the opportunity to apply for accelerated approval. And fortunately, in 2012, Congress sort of leaned on the FDA by saying, this is a recommendation. If you have a serious life-threatening disease with an unmet need in a rare disease population, we're going to tolerate accelerated approvals. And an accelerated approval allows you to use a surrogate endpoint that's reasonably likely to predict clinical benefit, or a clinical endpoint that can be measured earlier than irreversible morbidity or mortality that's reasonably likely to predict a clinical gain. And then the idea is that if you have data that looks like the drug is reasonably likely to work, you can get an accelerated approval, but the deal is that after the approval, there's continued post-approval registry where patients have to continue to be monitored. And if over time they were mistaken and the drug doesn't really work, they have the opportunity to take the approval back. But this will enable small populations of boys to participate in trials and get approvals when there aren't so many people affected by the disease. So in the case of rare disease, this is going to be a game changer because it's very difficult to get the number of patients needed to push through a conventional FDA approval pathway. So there's two ways in the context of Duchenne that an accelerated approval could go forward. It could be based on a surrogate marker, and the best surrogate marker here would be dystrophin. You've lost dystrophin, you're trying to replace dystrophin, this is the proximal defect of the disease. If you could show dystrophin is being made, perhaps that would predict clinical gain. And so there was a debate as to whether it could be used as a biomarker or whether or not the quantitation was robust enough that in and of itself it could act as a surrogate endpoint. And I think the consensus of the field right now is that by itself it's not robust enough at this time to enable approval as the only endpoint. But I think in the context of another endpoint, it should be very useful to bolster the data and may in fact end up being what makes or breaks the day in the end. The other possible mechanism for accelerated approval is to use the 6-minute walk test as a clinical endpoint that's reasonably likely to predict clinical gain, and then to follow with a post-approval registry. So this is sort of the package that eteplirsen is moving forward with the FDA. All of the data has been submitted, and the advisory committee meeting is just next week, January 22nd, and the decision for accelerated approval based on this phase 2B extension will need to be made by February. So just to remind you, the clinical efficacy was shown in that 151-meter advantage was found in the 6-minute walk test of boys who were eteplirsen treated relative to natural history controls. I didn't show you the data, but they've also measured pulmonary function and found that in these boys, over a period of time when you would expect them to lose a bit of pulmonary function, they have remained stable in their function. The biopsy, especially the 4th biopsy that was done in accordance to agreed upon protocols, demonstrated message by PCR in all patients. They also demonstrated positive staining in immunohistochemistry and by Western blot in 9 of the 11 patients. They rescored the original 2 biopsies, and 3 additional pathologists concurred with the first one, saying that in those 3 biopsies, all the boys were making dystrophin as well. The safety profile is excellent. There were no injection site reactions, no hospitalizations due to related adverse effects, and it continues to remain well tolerated after over 2,300 doses given. Tristan is the other drug in competition with Sarepta's eteplirsen. As I mentioned, BioMarin is now the owner of this technology, and they presented to the FDA Advisory Committee right around Thanksgiving. Their path was a little different. First, the drug was owned by Presensa, and GSK was helping to license it, and based on phase 1, phase 2 studies, GSK decided to do a phase 3 study, a definitive phase 3 study, and that study failed. And so BioMarin picked up the drug and decided that they would combine the promising studies from phase 1, 2, and 3 studies and ask for an accelerated approval based on the early results that seemed promising. So They too, of course, started with a short-term phase open label trial and showed, in fact, that Dystrophin, which then was called Pro-051, was able, after 5 weekly injections, to lead to the production of mRNA that was indicative of skipping, and they could show by protein and Western blot in these short-term studies that some dystrophin was made. During this early phase 1, phase 2 studies, a number of boys were treated and seemingly were stable for a while. And the fortunate thing is they had enough drug that they could keep these boys on drug for the following 177 weeks. And what you can see looks very much like the Sarepta data insofar as it would appear that those boys that were treated are stable. But this was an early study, there was no placebo group, and unlike the Sarepta data, the walk times at the beginning of the trial were not well matched. So you had a whole group of boys starting with different walk times and who had different ages and levels of progression. So that made it very difficult to use these data to match to a natural history control. But as I mentioned, since this drug was picked up by 2 different big pharmas, they were able to go through and do quite a bit of dose finding, dose ranging studies that hinted at efficacy. So they ran 2 phase 2 studies to examine this, and then, as I mentioned, those were sufficiently promising that they went forward with the phase 3 study, and unfortunately that failed. So you can see here that in one of the dosing cohorts, they had 50 patients, or 50-ish, and you could see that the folks treated with 6 mg per kg per week were able to retain their ability to walk better than the placebo cohort. Unfortunately, the intermittent group didn't look so good, so they decided instead that they would proceed with a full dosing relative to placebo. And in this study, they were able to see statistical significance at a couple of the points, but it wasn't consistent. You didn't see it at every point. At the same time, they were doing another phase 2 study aimed at establishing the best dose, either 3 mg per kg or 6 mg per kg. And again, at one time point, week 24, they were able to see statistical significance, but it was inconsistent in that the other time points didn't show any difference that was statistically significant, and that led individuals to question the robustness of the data. So as I mentioned, they went on to do a large 125 patient big phase 3 placebo-controlled study. And as you can see here, in this cohort, the drug appeared to have absolutely no effect whatsoever. It failed to meet its primary endpoints of walk time and dystrophin expression, and so the trial was halted, and all of the boys who had been on the drug over time were taken off. GSK dropped the drug, and over time, BioMarin decided that they would pick up the drug and go back to the FDA, not only with the failed phase 3 data, but with the phase 2 data. And they examined the phase 3 data better and came up with a hypothesis that perhaps it was the younger boys that were best affected. And if you could sort of cull the group just to the younger boys, you might be able to see some efficacy. Unfortunately, when you picked the group that worked in the phase 3, it was difficult to have that same group work in the phase 2 studies, and so there were inconsistencies between the studies. But you can see that in the first trial I told you about, there was significance at week 25 and week 49, and the difference in this case was a 35-meter difference on the 6-minute walk test. In the second study, they got a 27-meter difference in the 6-minute walk test, but only at one time point. And in the third study, there was no statistically significant difference between the groups. The FDA identified some problems with the data during the review of Dystrophin a couple months back, They were unhappy with the inconsistency between the 3 studies insofar as you couldn't stratify the data in one and apply that strategy to the other 2 data sets to yield statistical significance. Another problem is that they didn't have any pre-treatment biopsy, so it was very difficult to determine whether or not dystrophin was induced because we don't know if the boys had trace amounts before. But they were able to compare the treated group with the placebo group and look for dystrophin. Unfortunately, they had difficulty finding skipped RNA in all of the samples. They saw some in some of the samples. And the dystrophin protein expression was very difficult to see. In fact, they report dystrophin expression of 0.3% difference between placebo and control, but they also showed data indicating that that was within the noise floor of their assay. And then the fact that the efficacy was questionable made the fact that some of the safety issues were life-threatening more compelling and really led to a question as to whether or not the cost benefit was there. So these boys, 75% of them have injection site reactions. And the injection site reactions vary from being bothersome to being some that had boys leaving the study because they were so unattractive, so painful, maybe led to ulcers in the skin. Another major concern was proteinuria, and the urine to protein creatine ratio increased. But most concerning was the findings of serious adverse effects. 12% of the boys had serious adverse effects, and those included thrombocytopenia. That was the most common. 8 boys got thrombocytopenia, which can be life-threatening, and the unfortunate reality is that it wasn't something that presented early. As the boys were on drug longer and longer, more and more boys began to have thrombocytopenia. It appeared to be an autoimmune phenomenon where antibodies to platelets were generated and found in the blood. There was also a concerning event, which was an intracranial venous sinus thrombosis, which also left that boy worse for it, and he had to also stop the study. So at the advisory committee, patient families got up and testified regarding how efficacious they perceived the drug was, and many felt that it was working in their child. And that was the most compelling part of the advisory committee. But unfortunately, the advisory committee viewed this, the package as largely negative based on the questions that the FDA lobbied to the advisory committee. But in a sort of unprecedented moment, they didn't ask the advisory committee to vote whether they voted for approval or against approval, and rather simply asked, what do you feel about this aspect of the data, that aspect of the data? And every single time, the answers were such that they were not very enthusiastic. Nonetheless, the patient population has continued to petition that they believe it's working, and though the decision was supposed to be made in December, the FDA has deferred the decision to early January, and so we're going to be seeing both of these decisions coming out neck and neck. I find it interesting to look at the history of the drug treatment according to Sarepta's stock price. Sarepta is a small company, and this is its major drug. So unlike BioMarin or GSK, you can actually chart the progression of the trials by looking at the reactivity of the stock. What you can see is that in 2012, when they released the 36 weeks data on the boys, the stock shot up. It went further up after the 48-week data was released, and that increase in stock allowed them to raise the $125 million they needed to make enough drug to treat a large cohort of boys. Then, in 2013, the phase 3 of Dystrophin failed, and since the competitor failed, the stock shot up and stayed up until the FDA told Sarepta that they didn't feel they were ready for the NDA filing, in part due to this lack of convincing dystrophin data. And then, a while later, the FDA provides feedback to Sarepta as to how they could get accelerated approval. The stock went back up. The FDA accepted the NDA. The stock went up further. And then you can see that these two stocks are fluctuating greatly around the AdCom because it's anticipated that whichever of these companies gets approval, the market's worth $1 billion. So additionally, each company have follow-on exons. So if their first exon skipping drug works, they will take the same backbone chemistry targeted against a next mutation and begin to treat additional boys with designer medications. And BioMarin's already begun trials in exon 44, and Sarepta has already begun trials in exon 45 and 53. So the regulatory decisions for Dystrophin and eteplirsen are going to provide guidance to these pipeline drugs, but also to the entire field, because there never has been an FDA approval for Duchenne. This would be the first. There's a lot of drugs in the pipeline, and biotech watching to see what type of studies are going to be required for approval in the context of Duchenne, given that it's such a rare disease. So I hope to be able to update you in a final small lecture after the AdComs, and we'll keep our fingers crossed that drugs are approved for boys with Duchenne.